Ladies and gentlemen, I am Tuula Teeri, I'm the president of Aalto University, and I am very pleased to welcome you all to this seminar here at Aalto University. We have here all the distinguished delegates from the World Cultural Council. We have our distinguished award winners, and we have many colleagues and friends in the audience. And like I said, I'm really pleased to have you all here. When we are looking at the goals of the World Cultural Council, the admirable aim is to build and further relationships with and among prestigious scientific, educational, and cultural organizations worldwide, fostering cultural well-being, peace, and fraternity across the globe. Aalto University also is working toward a better world through excellent research and art, interdisciplinary collaboration, pioneering education, tangible societal impact, very importantly, and continuous renewal. We're striving to provide an academic and entrepreneurial environment where talented, passionate professionals and students from all over the world can connect and thrive. The missions of the World Cultural Council and Aalto University complement each other beautifully, I think. Aalto University seeks to produce new knowledge, technologies, innovation, expertise, and indeed experts that are able and willing to improve the living conditions of people. The World Cultural Council is recognizing such efforts of distinguished scientists, artists, and educators, making their achievements visible. So in a sense, both the World Cultural Council and Aalto University build on the well-known maxim often attributed to Sir Francis Bacon, scientia est potentia, I think, in Latin. Knowledge is power. So this insight of the nature of knowledge emphasizes that to acquire new knowledge means that one has the potential and the capacity, the power, to really make a difference. And it is very important how we use this power. It is a privilege, but it also is a serious responsibility. So the World Cultural Council's Albert Einstein World Award of Science is awarded for research which has brought true benefit and well-being to mankind. The educators receiving the José Vasconcelos World Award of Education are recognized for teaching the underlying basis of our present civilization, no less. And the Leonardo da Vinci World Award of Arts is for artists that are contributing to the artistic legacy of the world. They are all very noble and worthy goals for us. The understanding of knowledge as a capacity or indeed power to make a difference and to bring about well-being is also at the core of the Aalto University education strategy. The focus of our teaching effort is the student. Our aim is to educate responsible experts, future game changers who can lead change and renewal. Research-based new knowledge plays a crucial role in university education, but it is the student as a co-creator and our partner in learning that is in our focus. Ladies and gentlemen, last year I had the pleasure of attending the 2013 World Cultural Council Award ceremony at the Nanyang Technological University in Singapore. And in this event, Aalto University professor Petteri Nisunen and his colleague Tommy Grönlund were awarded the Leonardo da Vinci World Award of Arts. This highlights another parallel in the approaches of the World Cultural Council and Aalto University. With both of these organizations believe in bringing research, education, and art and culture together in creating a better world. So this year, Petteri Nisunen and Tommy Grönlund are presenting their newest creation of art by the entrance of the prize ceremony. It's a work of art that will bring beautiful light into the ceremony in the beginning of the dark Nordic winter, which amazingly enough today broke into bright sunshine. I'm sure in your honor. Well, I'm sure this piece of art as well in its part will make the world a little bit brighter for us during this prestigious ceremony that will be following. So dear friends, it's a great honor for Aalto University to host this World Cultural Council award ceremony. The award ceremony shall celebrate scientific and educational breakthroughs and achievements that have already proved their significance and that remain an inspiration to all of us. So we are very proud to welcome this year's laureates, the delegates of the World Cultural Council, and our many distinguished guests to Finland and Aalto University. So the program will now be launched by a historical account of one great scientific breakthrough from the distinguished World Cultural Council Honorary President, Professor Edmond H. Fischer. President Fischer will be introduced by Professor Jukka Seppälä, who will be chairing this session. Jukka is a professor of polymer technology and head of Department of Biotechnology and Chemical Technology at Aalto University. He currently holds the prestigious Academy of Finland professorship. So I wish you all a delightful, thought-provoking and enjoyable few days at Aalto University. And Jukka, please, welcome. Thank you.
Distinguished guests, fellow scientists, supporters of science and research, ladies and gentlemen, it is my great privilege and honor to present our opening lecturer, who is a role model and inspiring example to us younger scientists. Nobel Laureate, the President of the World Cultural Council, Professor Edmond H. Fischer. Edmond Fischer is one of the world's leading scientists. In 1992, Professor Fischer was awarded the Nobel Prize in Physiology or Medicine jointly with his colleague, Professor Edwin G. Krebs, for their discovery of reversible protein phosphorylation as a biological regulatory mechanism. Professor Fischer earned his PhD in organic chemistry from the University of Geneva in already 1947 and continued his research there until 1953 when he emigrated to the United States of America and joined the Department of Biochemistry at the University of Washington, Seattle. There at the University of Washington, Fischer and Krebs started their studies on the complex hormonal regulation of glycogen phosphorylase, studies that led them to discover that the enzyme was regulated by reversible protein phosphorylation. That is by the attachment or removal of phosphate groups. They called the enzymes that catalyzed those two reactions protein kinases and protein phosphatases. It soon became clear that reversible protein phosphorylation is one of the most prevalent mechanisms by which cellular processes are regulated. In contrast to the process of gene expression, protein phosphorylation is a very rapid way to adapt activity levels of enzymes and helps to fine-tune the protein activity. For example, the uptake of glucose is regulated by protein phosphorylation. In certain types of diabetes, these processes are disturbed, leading to increased blood glucose levels. Furthermore, protein phosphorylation is often a cause for the development of cancers. Professor Fischer has received numerous awards and honors in recognition of his contributions to science. He has been the president of the World Cultural Council since November 2007. The title today is The Origin of Reversible Protein Phosphorylation. Professor Fischer, please.
Can you turn the slide on? Thank you. Can you hear me? Is this machine working? Well, I'm delighted to be here, have this opportunity to speak with students and faculty at this university, and meeting many old friends, including a former postdoctoral fellow of mine who's seated there. He came, Professor Philip Cohen's traveled from Miami, New York, Helsinki all night, just listen to me. So at the end of my talk, I'll explain to you how these things work, Phil. Well, what I propose to do is to tell you sort of a very rapid overview of the work we carried out with my colleague Edwin Krebs on glycogen phosphorylase, a work that led to the discovery of protein phosphorylation as a regulatory mechanism. Now, Ed, who was a very old and dear friend of mine, died 5 years ago, so I introduced a couple of videotapes of him speaking while the two of us were just, chatting about our earlier work. The reaction that I will describe, reversible protein phosphorylation, this thing will not show on the screen. Anyway, this reaction is really embarrassingly simple. And when we came up with it, nobody would have paid much attention to it, except for the fact that it turned out to be crucial for the regulation of cellular processes. On the other hand, it's fair to say that when we came up with it, it came as a total surprise, because in those days one knew essentially nothing about cellular regulation, and things like signaling, signal transduction, words that are so commonly used today would not have been understood 60 years ago when we came up with that. And to put this in the proper context, let me tell you how this field was when we started working on it 60 years ago. First of all, while endocrinology was already well recognized as a discipline, it remained purely at the phenomenological level, mostly intact animal level. A few hormones were known that carried a message from one organ to the other and then resulted in some kind of a physiological response. So insulin was already known as the messenger sent by the pancreas, just like adrenaline was known as the messenger sent by the adrenals. But the action of the hormones stopped at the cell membrane. And then what happened next was absolutely unknown. It was inconceivable that a hormone could act on a cell-free system until Earl Sutherland and his student Ted Rall came up with cyclic AMP, which served as a second messenger, as an intracellular messenger of the action of adrenaline. Secondly, there was a very big difference in the way science was conducted in those days. In those days, well, in fact, since the days of Claude Bernard, the French physiologist who first described the action of the pancreas, one observed a physiological phenomenon and then tried to find the factors, the enzyme proteins responsible for it. Nowadays, by and large, it's the other way around. First, phenomena are described, and first proteins are described, and then by applying them, and so many have been, obtained by sequencing various genomes, by overexpressing them, by knocking them in and out, one tries to define their function. So to paraphrase Luigi Pirandello, we went from 6 functions in search of an enzyme to hundreds of enzymes in search of a function. And then, as I just said a moment ago, one knew essentially nothing about the way enzymes were regulated. The prevailing idea was that they were regulated by the rate they were synthesized and then degraded. And that stood until the maybe late 40s, early 50s, when it was realized that that couldn't be the case, that this wouldn't work, that protein synthesis and degradation were far too slow processes, that cells had to have ways of manipulating the activity of the enzyme once they had been produced and liberated inside the cell. They had to have the capability of responding almost immediately to external factors, adapt to the environment, satisfy their metabolic, physiological needs. Almost immediately, that had to be the case in response to whatever stimuli was placed upon them. And that was the big problem that faced the biologists at that time. And that was the problem that we decided to tackle with my friend Ed Krebs. We decided that we wanted to see how an important enzyme called glycogen phosphorylase was regulated. When I say important, in fact very important, because it was already known by the Coreys that it catalyzed the rate-limiting step of carbohydrate metabolism. That is, the first step, this thing, no, you can't see. The first step in the degradation of glycogen to give glucose 1-phosphate, and then finally blood glucose in the liver. And really, this is the most important function of the liver, production of glucose. And then energy in the form of ATP in every organ. Now, phosphorylase was a very attractive enzyme to work on because it was already known that it was under complex hormonal regulation. In the resting state, it is almost inactive, and just about nothing happens. But the moment you have a need to elevate blood glucose or to elevate your stores of ATP, for instance, if you are hypoglycemic or if you exercise and the stores of ATP down, it becomes instantly activated by the release of adrenaline. And adrenaline was already known to be released under conditions that the physiologist Cannon in 1920 termed action that will prepare an animal for fight or flight when presented with this sort of a emergency situation. In such a case, a signal is sent from the eyes to the brain, and the brain immediately orders the body to produce adrenaline, which is then transported in all the organs of the body. And you all know here what happens when you have a shot of adrenaline or an adrenaline rush. Okay. Heart begins to beat. In fact, it beats like crazy. Blood pressure goes up. Respiration increases. But if you have to fight or run away, the first thing you have to do in a hurry is to produce ATP. By contrast, if the stores of glucose or ATP are high, let's say after a big meal, the activity of the enzyme is inhibited by insulin. And in those days, insulin was already known to favor a deposition of glucose in glycogen. That was before Louis Leloir from Argentina discovered the so-called Leloir pathway with the involvement of glycogen synthase. And then approximately at that time, Christian de Duve isolated glucagon. So it was already known at that time that glucose homeostasis in the blood was regulated by the interplay of those various, by the opposing action of those various hormones. And of course, phosphorylase had to play a central role in those reactions. And that's why it was an attractive enzyme to work on. Here's a picture of Louis Leloir, taken at a meeting in Mexico City. You see the pyramid in the back, next to this good-looking young fellow. Glycogen phosphorylase was discovered in the early 30s by Jakov Parnas in Poland and Karl and Gerty Corey in the United States. And it was already known at that time that the enzyme existed in muscle enzyme, in a completely inactive form, and that it needed a compound, adenylic acid, 5' AMP, to become active. And of course, at that time, AMP was thought to be a coenzyme for phosphorylase, absolutely needed for the activation of the enzyme. And this stood until 1943, when a gal in Corey's lab, Arda Green, crystallized active phosphorylase. But she crystallized it in a different form, in that it was fully active without any need for AMP. So very logically, the Coreys assumed that in that new form, AMP was linked, they called it phosphorylase A, and it was linked covalently to AMP, that AMP served as what we call a prosthetic group. Furthermore, they assumed that that form had to be the native form of the enzyme in muscle, because if they took some muscle extract and let it stand for a while, it was rapidly converted to the old form. And they thought by an enzyme that would split the prosthetic group, an enzyme that they call prosthetic group removing enzyme. So an absolutely logical, straightforward hypothesis. But if that hypothesis were correct, AMP would have to be produced in the reaction. And they found none. Furthermore, using the most sensitive microbiological assays available at that time, they could find no AMP or adenine or ribose in the native enzyme. So they were stuck. They knew that phosphorylase existed in two forms, but they had no idea how those two forms could differ. And strange as it would seem today, they actually dropped that problem in 1943. This is a picture of Karl and Gerty Corey when they got the Nobel Prize in 1947. And here's another picture I took of Karl as we were hiking together around Mount Rainier in the vicinity of Seattle. Now, Ed and I became involved in this problem when we found ourselves in the same department of biochemistry at the University of Washington in 1953. Ed had been a postdoctoral fellow of Corey and assumed, like they did, in fact, like everybody did, that AMP was a coenzyme for phosphorylase. I had done my PhD at the University of Geneva, and among other things, we had isolated phosphorylase, but from potatoes. And it was fully active without any need for AMP. And even though biochemistry was an in its infancy, in those days, we knew enough to know that coenzymes were fully conserved among species. A coenzyme works in mussel, works in fish, and Drosophila yeast and plants. So it sounded absolutely unlikely to me that AMP could be a coenzyme for phosphorylase, mussel phosphorylase, but not for the potato phosphorylase, even though potatoes are pretty dumb compared to mussels. Okay? So we decided, well, why don't we take a crack at this problem? But Ed had some hesitations at first.
The fact that I had been in the Corey's lab and here just about maybe only 5 years later, we might start working on phosphorylate. And at that time, postdocs, when they left the lab, or students when they left the lab, did not work on the problem that the mentoring was going on in the lab. And then I got to thinking about it. 5 years since I was in that laboratory, certainly it ought to be permissible if we worked together at that time.
Does that work? Well, I can tell you right away that we never found out what AMP was doing. We had to wait 8 years for Jacques Monod and François Jacob and Jean-Pierre Changeux to come up with a very brilliant model, allosteric model of enzyme regulation. But what we rapidly found out is that there was another form, rather than allosteric regulation, another form of regulation, which was the hormonal regulation of the enzyme. Okay, so So we cleared the bench in a corner of the lab and started working, just one next to the other, much more like two friends than like two colleagues in a department. And the first thing we had to do, of course, is to prepare pure crystalline phosphorylase by the classical method of Corey. Method consisted in taking rabbit muscle, grinding it, extracting it in water for an hour, putting the gunk in cheesecloth, purifying the very, very turbid first extract by passing it through a battery of filter papers. And Gertie Corey always said you have to work very, very fast, otherwise active phosphorylase will be degraded to the inactive B form. So they poured the thing in that, and then with a big glass rod they went to the next filter and they squeezed it. It was an absolute mess. It was a totally archaic type of procedure. In fact, here's a picture of our lab. And Ed Krebs is here on the top hat. So we decided, enough with that jazz. Let's replace the filtration by centrifugation. In defense of the Coreys, it should be said that in the mid-30s, there was no good centrifuge available, no refrigerated large-capacity centrifuge. So we centrifuge our crude extract rather than filtering it. But as fast as we could work, as cleanly as we could work, we could never obtain active phosphorylase A, only the inactive form. So in desperation, we said, we have to go. It's crazy, it works for the Corey, why doesn't it work for us? We have to repeat Corey's procedure to the letter, paper filtration and all, measuring every step, every solution. And to our enormous surprise, this is what we found. That the very first extract we got from mussel was not active phosphorylase as we expected. but the inactive form, but when it passed through filter paper, it was converted to active phosphorylase A. Now, speaking of a disappointment is really to put it mildly. You know, two young fellows thinking that we'll find a new mechanism, a new chemical reaction, maybe a new vitamin, a new factor. Paper filtration to activate an enzyme was really below the worst. You know, it was the pits. To such an extent that we did not believe that we were really dealing with phosphorylase B. We thought it must be an inhibited form of phosphorylase. Perhaps it's bound to subcellular elements, a big complex, which maybe is removed by the paper. So, to such an extent that in our lab books, we referred to that fraction as false phosphorylase B. But then it turned out that no, it was real phosphorylase B, but what happened during the paper filtration was not the filtration per se, but the fact that in those days, all filter papers were contaminated by traces of calcium ions. And enough were picked up by, in those 20, they could use 15, 20 filter papers in their procedure. It was the calcium that was picked up. If we, for instance, pre-washed the paper with dilute acid, no conversion. Finally, what's clenched it is when we took about 20 of those filter papers and ashed them in a muffled furnace. And then rather than filtering, we simply spread the ashes, and it worked. And then very rapidly we could find that calcium did not work alone, but needed also ATP. So the reaction was a calcium ATP requiring reaction, and this immediately suggested that we were dealing with a phosphorylation reaction. So of course we had no idea what had become phosphorylated, because in those days you had no ATP32. You had to synthesize the damn thing yourself. But we knew that Arthur Kornberg, who was in St. Louis, had prepared some ATP32 that he needed for the beautiful work he did on biosynthesis of DNA. So we called Art, we used to call him King Arthur, we called Art, and he immediately sent us some radioactive ATP, gamma-labeled ATP32, and with that compound we could show that radioactivity was introduced in a protein fraction, which we could purify and which turned out to be phosphorylase. So for the first time, we could report, publish, that the activation of phosphorylase occurred like so, that inactive phosphorylase B would become phosphorylated and activated by an enzyme which we call phosphorylase kinase, in the presence of calcium, magnesium, ATP. Now, what would seem today as a, very simple, if not completely trivial reaction, came as a very big surprise. Very big surprise because in those days one knew essentially nothing about phosphoproteins, and the idea that a phosphorylation reaction could serve as a mechanism to activate an enzyme was totally unbelieved. In fact, this is how phosphoproteins were described.
One of our very first experiences, the editorial board of the Journal of Biological Chemistry was very small. And I think there were 5 or 6 people in the country who reviewed all of the papers that were submitted to the Journal of Biological Chemistry. And I do remember that when Ed and I sent in our first paper on phosphorylase, we were aware of the fact that
Carl Corey
was one of the 5 or 6 editors of the Journal of Biological Chemistry. And so when we sent in our first paper on phosphorylase, we knew that he would see it. And we had faith, which was justified, that he would not take advantage of seeing that paper, and he didn't. He himself was very excited by our findings.
So, as I was telling you, it's absolutely trivial reaction, but in those days one knew nothing about phosphoproteins, and this is how they were described in the Hawke, Ozer, and Summerson in 1955. That was a classical source of biochemical information. These proteins contain phosphate bound in ester linkage to the hydroxy amino acid serine and threonine. They include casein from milk, ovovitelline from egg yolk, and other proteins associated with the feeding of the young. They were devoid of any biological activity, considered totally uninteresting, and in fact dismissed as non-entity at that time. So the big surprise. But then the reaction I just showed you became a little bit more complicated as soon as we begin to carry it out with partially purified elements. Oh, we had crystallized phosphorylase B. We had purified a little bit phosphorylase kinase. And when we carried out the reaction with purified product rather than with the crude extract, it was certain, clear, that calcium could not be involved in the reaction. And yet, it was absolutely required if you work with muscle extract. If you had a crude muscle extract, you will find that there is a lot of phosphorylase B, a lot of phosphorylase kinase, a lot of magnesium ATP, but no reaction takes place before you add calcium. So the only possibility was that calcium had to act at an earlier step. And the possibility that phosphorylase kinase also existed in an inactive and active form, and maybe calcium was required for that reaction, and that turned out to be correct. In the presence of calcium ion, there's a rapid phosphorylation and activation of phosphorylase kinase, which can then activate the phosphorylase reaction. So already there we had a sort of a cascade of one enzyme acting on another enzyme, resulting in the breakdown of glycogen. Note that at the same time, Earl Sutherland and Tedroll had arrived essentially to the same conclusion. They were working not with striated muscle, but with liver phosphorylase. And they showed that when active liver phosphorylase was incubated with another liver fraction, inorganic phosphate was released, implying that it had to be a phosphorylation, dephosphorylation reaction. But an epochal finding that grew from those studies was the discovery by Earl of cyclic AMP, which by a totally unknown mechanism shifted the equilibrium between those two forms to active phosphorylase A. Earl was a good friend. We saw him at every Federation meeting. We discussed his work, our work. Late at night, did a lot of drinking, which finally did Earl in, unfortunately. But Earl immediately sent us a fraction of cyclic AMP, and we could show that its action was directed towards this step, the activation of phosphorylase kinase. We didn't know at that time whether or not all it did was to accelerate the autophosphorylation of an enzyme, it's a protein that could phosphorylate itself, or perhaps that it could act on yet another enzyme. which, for want of a better term, we called a kinase kinase. And some people thought it was a funny name, other people thought it was a stupid name. And Ed and I remember very well that the reviewer, JBC reviewer, who reviewed that paper, first of all, he did not believe that there could be another enzyme. He wrote, "When will it end?" But secondly, he wrote, "If indeed another enzyme is involved, I wish the author would find a better term than a kinase kinase." I wonder what he would have said if he had known MAP kinase pathway, where you have MAPK, MAPKK, MAPKKK, all acting successively on one another. Anyway, that hypothesis was confirmed 4 years later by the isolation by Ed Krebs and Don Walsh of the cyclic AMP-dependent protein kinase. And then since at that time Earl Sutherland had elucidated the formation of cyclic AMP at the membrane, the entire cascade for the phosphorylasis of glycogen was established. Now, we were very interested in the involvement of calcium in that process because the physiologists had known for many years that calcium released from a nerve impulse could bring about muscle contraction, activation of thin filament, formation of actomyosin. The possibility that calcium could also affect carbohydrate metabolism was really bizarre. And to show that those two reactions were synchronized, we had to show that the same concentration, very low concentration, needed to bring about contraction would bring about the activation of phosphorylase kinase. And that was done in collaboration with a guy from our physiology department, Glenn Kerrick. He told single muscle fibers free of their membrane and attached to a tension transducer. Since those fibers lacked a functional sarcolemma, a functional membrane, the ionic environment could be strictly controlled while isometric tension could be measured. And we could show that as contraction increase as a function in increasing concentration of calcium, phosphorylase kinase is also activated. And the finding that the same concentration of calcium that triggers contraction would activate indicated for the first time how two separate, different physiological pathways could be activated. Muscle contraction and then activation of phosphorylase kinase, production through glycolysis and Krebs cycle, formation of ATP needed to maintain contraction. We did not know at that time whether or not that phosphorylation reaction, the activation of kinase and phosphorylase, were restricted to only those enzymes, maybe one or two other enzymes of glycolysis. We wondered, for instance, if the enzymes involved in nitrogen metabolism could be activated by amidation, deamidation. The enzyme of fat metabolism might be activated by acetylation, deacetylation. And as you well know, it so happened that phosphorylation became one of the most prevalent means of control of cellular processes, being involved in the control of metabolism, transcription, translation, the immune response, differentiation, transformation, cell death, and many, many pathologies or hereditary disease. It's directly involved, of course, in diabetes, but also in Alzheimer's, Parkinson's, myelogenous leukemia. It directly involved in bacterial disease such as the plague or cholera, in viral diseases such as smallpox. In fact, it would be very difficult to find a physiological reaction that is not directly or indirectly regulated by a phosphorylation reaction. Now, better than 99.9% of all those reactions occur by phosphorylation of those proteins on serine and threonine. But one of the most exciting developments in this field was the discovery by Tony Hunter at the Salk Institute, working on House sarcoma virus and polyoma middle T antigen, that tyrosine phosphorylation of proteins on tyrosyl residue was intimately implicated in transformation and cancer, involving a whole bunch of viral or cellular tyrosine kinases, or tyrosine kinases belonging to growth factor receptors. As a matter of fact, it was found that the plague, that the virulence of the bacteria, Yersinia pestis, the bacteria responsible for bubonic plague, that the Black Death, that wiped out a big proportion of the human population in century past, result from the fact that the bacterium injects in the host cell a tyrosine phosphatase, nothing else. And this tyrosine phosphatase brings about a catastrophic set of dephosphorylation, which totally wipes out the immune defense of the cell. If that phosphatase is removed from the bacterium, as done by Jack Dixon, the bacterium is totally harmless. Can you imagine that the Black Death is due simply to one enzyme, a phosphatase, a tyrosine phosphatase that is injected in the host cell? Okay, now I would like very rapidly to come back to this business of AMP. And as you know, AMP were found to be an allosteric effector of phosphorylase. But the question is, why did organisms found it necessary to have two types of activation? One, an allosteric activation. On the other hand, a covalent phosphorylation of the protein, when in both cases you end up with an active enzyme. Well, according to the classical model of allosteric regulation by Monod, Jeffrey Wyman, and Jean-Pierre Changeux at 65, proteins will change their conformation in response to allosteric effectors that are liberated inside the cell during the normal maintenance of the cell. Depending whether or not the cell is actively reproducing or quiescent, actively metabolizing or not, its energy charge, the ratio of AMP over ATP with ADP plus ATP, its redox state, etc. According to the general rule that enzymes are inhibited by the product of the reaction, feedback, end product regulation, phosphorylase would be expected to be inhibited by G6P, rapidly produced during its reaction, and it would be expected to be inhibited by ATP, the end product of carbohydrate metabolism. And likewise, it would be expected to be activated by AMP. which indeed it is. But now many enzymes of glycolysis of the Krebs cycle, they are all activated by AMP and inhibited by ATP. So if you relied only on that system of regulation, you would have to affect all those enzymes simultaneously, unless you had strict compartmentation. Whereas covalent regulation is enormously specific. Phosphorylase is activated by phosphorylase kinase only, no other kinase. So it allows you to open up that step, the breakdown of glycogen, without touching any others, opening that door without affecting any other. We were enormously lucky to have chosen this enzyme, this system to work on. First of all, phosphorylase is very abundant in muscle, so material was never an object. Secondly, the reaction is unambiguous, converting a totally inactive protein into a totally active one. Thirdly, it's stoichiometric, introducing exactly 1 mole of phosphate per mole of enzyme subunit. And finally, by luck, this phosphorylation event occurs on close to the N-terminus, serine 14, as discovered by Phil Cohen. So you could easily cleave off that peptide, leaving much of the protein behind, which simplified the isolation of the phosphopeptide, even though in those days it took a long time because there was no automatic nothing. There was no ionic, hydrophobic chromatography, no HPLC, no Edmond degradation, no, of course, MS, etc. Everything had to be done on little separations of peptide, on little pieces of paper by chromatography or electrophoresis, sprayed with anhydrine. Everybody had blue fingers in the lab. But then we could find out that there was activated, a single serial residue had been phosphorylated. And as I said, Phil discovered which one it is, very close to the N-terminus. That reaction was so straightforward, so simple, that there was no question in our mind that it would represent the prototype for those kind of reactions. As it turned out, it's probably a unique exception. Do you know other system activated by a single phosphorylation site? Myosin, yeah, he's right. Well, it took, I think, 6 years for Joe Larner to find the third enzyme regulated by phosphorylase, glycogen synthase. This is the enzyme of Louis Leloir. And Joe found that it inhibited the enzyme rather than activating phosphorylase. And that makes sense, because closing the backward reaction, the synthesis of glycogen, when the forward was activated, prevented a futile cycle resulting in a useless expenditure of ATP. But Joe rapidly found that far more than one phosphate was phosphorylate. 2, 3, 4, 6. He was always on the telephone. At that time, the idea that a single phosphorylation event was all that was required to activate an enzyme was so ingrained that I remember we were thinking that maybe glycogen synthase contains 6 identical subunits, all phosphorylated on a single site. And Joe also thought that. Now we know from his work, and mostly from work of Phil Cohen, that whereas phosphorylase is activated by a single phosphorylation on a single site, a single serial residue, glycogen synthase is hit by no more than 8 kinases, all totally unknown in those days, 8 kinases on at least 9 different phosphorylation sites. And we know that this phosphorylation requires the concerted action of several of those enzymes that trigger a complicated program of serial reactions that have to proceed in a precise order. From the work of Phil, we know that the synthase is inhibited by glycogen synthase kinase 3, an enzyme that he discovered, who hits the enzyme towards the middle on 4 serial residue. You had it there. But no phosphorylation can take place until a different enzyme, casein kinase 2, introduces a first phosphate downstream. Only then can GSK3 take over and phosphorylate the next residue, then the next, then the next, then the next. If that first serial is mutated, as done by Peter Roche at Indianapolis, if that serial is mutated, or that one is mutated, no further phosphorylation can take place, no regulation of the enzyme occurs. Now, imagine the incredible mess we would have been in with Ed if we started working on that enzyme rather than on phosphorylation. We would have never, but absolutely never, been able to resolve that problem. We were often asked with Ed if we realized when we did this work that we were working with a very ubiquitous and therefore very fundamental reaction. Absolutely not. We knew it was a nice reaction, we knew it was an interesting and important one, and this is why we stayed with it, but we never could have guessed all the events all the development that occurred after that. In those days, in the mid-60s, late 60s, everybody was thinking in the terms of allostery. At meetings on protein, the big question was, is this enzyme modified by the induced fit model of Dan Koshland or the allosteric model of Monod? And it was very clear to me that Monod, who was a very close friend of mine, that's a picture I took of him in Seattle once he was visiting, that Jacques never believed 1 second that covalent modification by protein phosphorylation was really involved in basic cellular. He simply didn't believe it. With his death 40 years ago, and then death of Danny Koshland, shown in the left there, I think 7-8 years ago. This is Sir Hans Krebs. This is the fellow who discovered the Krebs cycle. And P.P. Cohen in the middle. And then with the passing away of Ed Krebs just 5 years ago, we really have witnessed the end of an extraordinary era. Thank you for listening.
Thank you, Professor Fischer. I think, I'm
if
sure.
If you want to call me Professor Fischer, how about Herr Geheimrat Dr. Professor Fischer? Otherwise, you call me Eddie Fischer, like everybody else.
I got that advice from you when we discussed. Thanks, Eddie. So I said as introduction that you are a role model and inspiring example to us younger scientists. I think this proved that. Now it is excellent opportunity for us to discuss of your career and about this lecture. The topic itself, that is difficult, but I know there are experts. Please, first question is here already.
I know one here. Phil, I can explain to you how this thing works.
My name is Carl Gamber, I'm a biochemist. We have 500 kinases in the human genome and 100 phosphatases about. You can imagine that how complicated this will be. Do you think that we ever can really solve, let's say, a complete phosphorylation events in cells?
I think there are about 500 kinases. Phil can probably tell you the exact number, and about a little more than 100 phosphatases. So maybe less than 700 genes, I think, involved in the regulation by phosphorylation. But then the guys working in ubiquitination came. There are probably more than 2,000 genes involved that kind of regulation, and Phil has switched to this area. Yes, and then there are the serine-threonine phosphatases, there are the tyrosine phosphatase, there are the receptor form, the non-receptor form, and then the dual specificity phosphatase that can hit both serine, threonine, and tyrosine.
Can we ever understand this, how it really works?
So,
Will we ever understand how a cell really works when you think about the phosphorylation?
well, you know, if I understand your question correctly, it was thought maybe 50 years ago, 60 years ago, that all the regulation was through the kinases, that the phosphatases were just there to remove the phosphate introduced by the kinases. We know now, and I say mostly by the work of Phil and his wife Tricia, that the regulation of phosphatase is much more complicated than that of the kinases. They need targeting proteins. They need a whole series of different complex formation. So the whole field is one of course, great interest to the pharmaceutical companies because they are working on those enzymes now to cure a variety of diseases. But it's not an easy problem. When Tony Hunter found that he had found phosphorylation on tyrosine linked to house sarcoma virus, which it was the SARC gene, the gene responsible for the oncogenicity was a tyrosine kinase. He called us in Seattle, and we thought with it, we'll solve cancer within a year. And there have been very few cases, Gleevec, imatinib for chronic myelogenous leukemia. But I think we have great hopes, I certainly do, and I'm sure Phil has too, that sooner or later we'll be able to target those enzymes to cure a variety of diseases. But it's not an easy problem. It doesn't happen but it will.
Other questions? On my behalf, I would, of course, having looked on your CV, it became clear that originally, as young scientist, you were organic chemist, and then you moved to biological chemistry, and in the lecture also it was clear that it was early stages of biochemistry and biological chemistry at that time. What was the reason? Why
to
did
tell you the truth, I wanted to become a microbiologist. Yeah. I had a huge admiration for Louis Pasteur. My father had tuberculosis, and as a kid I wanted to become a microbiologist, maybe trying to find a cure. for he was a chain smoker and he died of it much before I could enter the field. So when I finished school, college, which we called, I went to see the chairman of microbiology in Geneva to ask him his advice as to how I should proceed. And I remember Fernand Chaudat was his name. And I always remember what he told me. He told me, you know, if you want to become a research bacteriologist, you should study chemistry first. He said, nowadays we use test tubes much more than we use microscopes. So this is why I entered the School of Chemistry in Geneva and got a PhD degree in organic chemistry, but I always had an eye on the biological aspect. This is why my thesis was on enzymology, isolation of an enzyme, etc. So, it was a very good combination. Ed Krebs was a MD, you see, and I was more of a chemist. So it worked out very nicely.
So it was your passion towards life sciences.
Yes.
What would be your advice to young scientists thinking about their orientation and future areas?
They should do what passion they have. They should do really what they want to do, whatever it is. Because you cannot say that one particular field has any advantage on any other. Just like, you know, young biochemists, they come to us, to me, they say, you know, what project should I work on as a young assistant professor? And I tell them, you know, whatever project that really excites you, that you like. Not to choose a trivial project, to choose a very important one, but one that is not too much worked by other groups. You cannot compete with a pharmaceutical company or a guy who has 30 postdoctoral fellows. So, éviter les plates-bandes trop piétinées, as you would say in French, to avoid the flower beds in which too many people are working on, but mostly something that excites you.
On Friday night, when we drove from the airport to the hotel, we already then discussed about progress of biosciences, biochemistry, and you mentioned certain exciting areas of today's visions for the future. Could you define what excites you most today when you look forward?
What one doesn't know. I'm much more interested in the mysteries of science than what science has taught us, even though, of course, it's impressive what it has taught us over the centuries. But I'm interested in all the problems that are yet to be solved, in physics, in astrophysics, in anything. You know, that I'm retired, I read Nature, Science, the scuttlebutt you have in front, little scandals that happen. But I read, that's the advantage. Once you are retired, once you are actively involved, you have to read certain journals and certain papers. And you have to remember what you read. And you have, every time, you know, half of the time I read a paper, I say, oh, geez, I should tell Al, we should do this, we should try that, we should try that. Once you're retired, you don't give a darn about that. You read whatever paper is of interest to you. I read a paper in, I don't know, quantum mechanics. I don't understand a word of what they say, but I'm interested to know why are they asking that question? I mean, what are they trying to find? And that's perfectly satisfying to me. So,
but your own field, I mean, in biosciences, we mine more and more data. Has it become more difficult, more complicated, more like information dependent? Or how is it? Is it
yes.
Do we solve problems or are we more confused?
You are right, but you can't do anything against that. When I went to America and started working in enzymology in 1950, I can assure you that I read every paper that was published in the field of biochemistry. The Journal of Biological Chemistry was a small format like that. There were maybe 50 pages per issue, published once a month. Today's about 30,000 pages. So this is an explosion that has happened, and there were very, very few biochemical. Nature at that time was considered like a joke. Nobody read Nature. It was a terrible journal. And now it's one of the most difficult ones, the most selective one in terms of publishing paper. And the literature before the war was 80% in German. You had die Berichte, the Annale von Biochemie, all the books were German books when I did my studies. There was no biochemistry book in French. There was a Polanovsky, which was terrible. There was a couple of good British books. When I went to America in '53, I realized I didn't know biochemistry. The textbook was thick like that. And that didn't exist in Europe. Now, of course, it's all over the world, and the mix. And I would say that 95% of the literature is really in English. And at meetings, in fact, like here today, you speak English. So it has changed considerably, but you can't prevent that. When we isolated the first tyrosine phosphatase, a student who got his PhD degree was Phil, and then he came to me as a postdoctoral fellow, a very bright fellow by the name of Nick Tonks. And we found out that this tyrosine phosphatase was part of a receptor, very, very well known by the immunologists. So I had to learn something about immunology and the immune response. When I started, there were maybe one volume in immunology, a journal. I think there are more than 50 today. So that tells you how science is going. So they become much, much more specialized, and it's much more difficult. I think for the young investigator, they have to concentrate it on their particular area. On the other hand, they have to learn about the whole field. Fortunately, today you have seminars, every week, every department, microanatomy, physiology, pathology, biochemistry. Every week you have a seminar. So you could spend all your time going to seminar to learn something. So difficulty is not to listening, but to remembering what you...
We have one more question from the audience, please.
Yes, thank you for a very inspiring lecture. Concerning your reply to the previous question on what is your advice for young scientists, you said to focus on your passions, and I completely agree with that. You also said to find your own niche and not to work in an area which is overcrowded and too competitive, and I agree with that too. I think there's a problem though. I tend to see that in most countries there is a tendency of funding agencies to want to support more applied areas of research, and that, first of all, I think is a bad thing for research in general, and it certainly does not help young investigators to follow their passions. Sometimes there has to be some level of compromise between what we are passionate about and what we can actually do with the resources that are made available to us. So I don't know if you have any views on that and any suggestions on how to counter this trend, which I find that is very preoccupying and that is happening pretty much worldwide as far as I can tell.
You are Institut National de la Recherche Scientifique. Did they pay your salary? They pay all your demands, or what?
Doesn't go much further than my salary.
Yeah. So yeah, this is a system we do not have. You see, in America, we do not have like they have in France, CNRS, or Max Planck in Germany. We don't have that. We have now the HHMI. They have their own laboratories, but very few people can be supported by them. You have the question?
There's one more question, please.
Yes, can you tell us why and how nature select phosphorus for that purpose?
Why when...
And how nature select phosphorus for that action?
You see, people often ask why a phosphorylation reaction? Because the bond is very stable. It has many forms of ionization. And at neutral pH, those bonds are very, very stable. Even the pyrophosphate in ATP, for instance, if you can, at pH 7, you can keep it for a year, it will not hydrolyze. And people wonder why they use ATP rather than, let's say, acetic anhydride, which is much more energetic. Acetic anhydride hydrolyzes immediately in water. I think that's because of that. It's a very abundant, like why in brain one of the principal transmitters is glutamate, because glutamic acid is very, very widespread. So I think that is one of the reasons. People have wondered, in the primordial times where the Earth was in a reducing atmosphere, if before phosphorylation they had maybe hypophosphate or some lower degrees of oxidation, they have never found any evidence for that. Even the obligate anaerobic bacteria that you find, in the sea, the other obligate anaerobic bacteria, they will utilize ATP and not and phosphate and sulfur mostly. So I would think that this is the reason. It's a very common, easily accessible, and forming very stable compounds.
Thank you very much.
I don't see any more questions from the audience. If I would try to conclude something which is, well, scientifically it's difficult for me, but in any case, it's clear based on what we have heard that phosphorylation is a very central mechanism in life and has effect on so many like diseases and functional life science features that it's incredible. Also, it is clear that we speak of so complex field where modern technologies progress in those, data mining, analysis, gene technologies, and synthetic biology in future may and surely offers enormous visions for the future. And another dimension that we discussed here is that we should follow our passion. Passion towards welfare is the driver to solve some of the life's great problems and to improve the world. I understand that has been the driver also in your career.
Well, in the field of phosphorylation, 90% of the development is due to the work that Phil has done in Dundee. And we could have never guessed those developments, never.
So finally, I only say that please join me to thank Eddie for his last...
Now you get it. Thank you.